Bonjour, mesdames, messieurs. Bienvenue à ce premier petit déjeuner avec des tata papineaux en ligne pour cette session parlementaire. Before we begin today's session, I'd like to begin by acknowledging the Indigenous peoples of all the lands we're on today. Although we meet on a virtual platform, we acknowledge the importance of the land which we each call home. We do this to reaffirm our commitment and responsibility in improving relationships between nations and to improving our own understanding of the local Indigenous peoples and their cultures. From coast to coast to coast, we acknowledge the ancestral and unceded territory of all the Inuit, Métis, and First Nations people that call this nation home. We pay respect to the traditional knowledge keepers, both young and old, and honor their courageous leaders, past, present, and future. Ladies and gentlemen, on behalf of PAGSI, which stands for the Partnership Group for Science and Engineering, it's my pleasure to welcome you to this first online Bacon and Eggheads event. I'm Natalie Goto, Chair of the Bacon and Eggheads Committee. For those of you who don't know us, PAGSI is a registered not-for-profit umbrella organization of 21 national science and engineering societies representing more than 50,000 individual scientists and engineers from academia, industry, and public service. This year, we're celebrating our 25th year as an organization, the last 20 of which have been involved in the organization of Bacon and Eggheads lecture series as one of our more public roles. While the current pandemic-imposed restrictions prevent us from meeting in person over what's normally a mouth-watering selection of breakfast goodies on Parliament Hill, we're pleased to be able to bring you the most important element of our breakfast, which is our egghead, which is the term that we affectionately give to our outstanding Canadian engineers and scientists who come to present their innovative research to parliamentarians and decision makers. We hope everyone will enjoy this opportunity to hear firsthand about the excellent research being done in Canada and take this opportunity to ask frank questions about the implications of this research for policy and future actions. All members of our audience will be able to submit their questions at any time during or after the talk using the Q&A panel within Zoom. You should be able to see a little Q&A option in your Zoom control panel at the bottom of your screen. And if you click on this, you can type in your questions at any time. Our speaker will be given an opportunity to respond to your submitted questions at the end of his talk. Before I turn the platform over to our introduction, it's important to tell you that the Bacon and Egghead series is generously supported by a number of organizations, which were shown in the slides during the waiting period. We're grateful to all of them, including the International Development Research Centre, NSERC, Juno Canada, and Laval University. We'd also like to thank our patrons, the Speaker of the House of Commons and the Speaker of the Senate. Thank you for your support. At this point, I'll turn the platform over to the President of the International Development Research Centre, Dr. Jean Lebel, who will introduce our speaker.
Thank you, Natalie, for this introduction. Deputy Speaker of the House of Commons, members of Parliament, distinguished guests, and we are a large audience today. Thank you for being with us today. Bonjour à tous. Merci d'être avec nous. It's a pleasure for me to introduce Gary Kobinger. It's a pleasure for me also to participate in the first virtual events of the Bacon and Eggheads. It's because of my French pronunciation of the H that I'm using the image. I'm the president of IDRC. IDRC, the International Development Research Centre, is a crown corporation of the Canadian government, part of the foreign affairs and development efforts of the country. We champion and facilitate research on the challenges that developing regions of the world are facing globally, and the COVID situation demonstrate that global is also local. It is for me an honor and a pleasure to introduce Gary. Gary and I have known each other for some time, and what a better way of informing parliamentarian as you, the audience, by having some of the most brilliant person in vaccine development in this country participating into today's event. Gary and I have been over the year interacting. For some of you, it might be known, but at the time of the Ebola crisis in West Africa, it was the leadership of PIAC and Gary, the Canadian Institute of Health Research, the Global Affair Canada, and IDRC together that gave the opportunity to test the Canadian vaccine that Gary worked with his colleague at PIAC in Winnipeg, that gave the opportunity to do the ring vaccination in Guinea. The ring vaccination that proved to provide a cure for this sanitary emergency. That was a Canadian story. That was a Canadian story with many actors worldwide, and that demonstrate the true value of collaboration of science. Collaboration these days is more than ever required. The COVID pandemic shows that this worldwide situation is affecting everyone drastically. The collaboration is required to develop rapidly and urgently vaccine of cures. But it is also important to remember that this global effort needs to be taken into consideration also with other aspects, whether it's access to this vaccine when it's ready, whether it is dealing with inequity in health services in order to provide the provision to the population that are the most affected about the access and the delivery of the vaccine. It is also about dealing with the information and disinformation. Science brings value in responding to those issues. But today, more to the point of our conversation, Gary will give us a bit of his view and experience of dealing with the development of vaccine for pandemics and contrasting the experience on Ebola as well as the one that he is leading now on the COVID. It is an unprecedented challenge, and Canada has much to offer as he will present through his own experience. Whether it was the years that we worked together in West Africa, whether it is the year that he's currently working at Laval University as a professor and director du Centre de Recherche en Infectiologie and Canada Research Chair in Novel Vaccine and Immunotherapy, Gary remained a person that is extremely knowledgeable, but that is also extremely humble. Throughout his career, whether at Laval or whether at the National Microbiology Laboratory in Winnipeg, Gary has shown the humbleness of scientists. Looking at difficult questions, working long hours, and empowering people around him to do the work with him. As Bocuse was saying, in order to be truly a great chef, you need to know how to do it. You need to let others work with you to be able to do it. And you need to let the world know how to do it. And this is what Gary will do today. As a friend of many of you, as a personal friend of I, but as one of the leaders in the world of vaccine development, it is truly an honor and a pleasure to introduce one of the most prominent researchers worldwide on vaccine development with innovation, creativity, but also humbleness. Gary, the floor is yours. It's a pleasure to listen to you. Thank you.
Thank you very much, Jean, for this very generous introduction. I wasn't sure if it was really me you were talking about at some point. I hope that the sound is good. You can hear me. I'm gonna share my screen so that I will start with my slides. First, telling you that it is an honor and a great pleasure to be with you today and have the opportunity to present on such an important subject as vaccine in normal days for me, but even more so in our current situation, facing obviously the ongoing epidemics of COVID-19. I would like today to talk to you about innovation to counterbalance challenges before, during, and after outbreaks, and highlight how actually contribution, including Canadian contribution in the past, are being important today. And we hope certainly, and I am convinced for the future as well. So we'll find a way to switch this. I will start with my conflict of interest statement. I've worked on many vaccine platforms. They're listed there. I've also worked for clinical development of some vaccine in phase 1 mainly, but 1 or 2 in phase 2. And you see that the pathogen listed here and the company that the work was done with. I've contributed my team of the development of ZMAP also towards the end with the collaboration of a company, and I've received honorarium as many other scientists from university that I give back to grad students. And I'm the funding president of the not-for-profit Guardrx. I'm going to touch on it towards the end of this talk. I will start by showing you this and telling you that while we are certainly very much aware of how an emerging virus can do damage to public health, to the life of citizens of all countries worldwide, and the economy worldwide, these are event that keeps coming back, and we have several examples. Even if we have an unfortunate tendency to forget them, some of them we are marked by, including, for example, in 1918 Spanish flu or influenza that did a lot of damage at that time too. But there is a lot more than that, and this is just to highlight the importance of being prepared for those events and to continue to develop the tools, including vaccine, to be able to minimize the damage of those events. This slides, aside from the cost of life, the cost of public health, the burden on public health, there is an enormous economic cost to those epidemics, those emerging and re-emerging infection. And you don't need to see all the details of that slide. What's important is that big blue, that big yellow, for the one that are not colorblind, that big yellow bubble, is the cost of the SARS epidemic at the beginning of the 2000 years. We had cases here in Canada. Overall, globally, that was less than 10,000 cases, and the estimate cost was $40 to $50 billion. Now, COVID, as of today, as written here, would be 100 to 1,000 times bigger than the largest circle, which is the yellow one, and would be way bigger than the slides. So I did not include it for obvious reason. One of the tool, if not the tool, that has been in relation especially to the cost of this preventive measure, and I'm talking about vaccine. Vaccine has been the one medical intervention that has saved the highest number of life in the history of medical science. The second, it is second only to another intervention, which is clean water. So vaccine is a very important tool to prevent disease, a very important tool to stop progression or transmission of an infectious disease. And on this slide, you see a long list of vaccines that were developed starting in 1797 with the first vaccine, which was a smallpox vaccine. And what I want to highlight with what's in red towards the bottom is we are just starting now to develop new tools in vaccination, tools that we call mostly experimental vaccine. And we are in the midst of developing a lot of and using a lot of those experimental platforms against COVID. Vaccine do take time. This is a list of pathogen from discovery and how many years on the extreme right column, how many years did they take to develop a vaccine, some of which we still don't have a vaccine against, including Zika, including RSV, respiratory syncytial virus that affects, including in Canada, the less than 5 and also the elderly severely every year. This slide now, thankfully, I need to update it because there is now a licensed Ebola vaccine towards the end, but it shows you how many years it takes. It takes a lot of time. This is why now when people say it takes too long for a COVID vaccine, in reality, we're trying to compress 20, 30, 40 years sometimes of vaccine development into this 1 year or 2 years from discovery of a pathogen. So it is an extremely big challenge that comes and thankfully is meeting with a lot of coordination, a lot of strength that we had in place and that we are continuing to accelerate. And this slide, I wanted everybody to just visualize when we talk about clinical progression, this phase 1, 2, 3, where do they sit? Well, you see from the concept vaccine, preclinical research, which is mainly testing in labs, including in animal models, that is a necessary step before entering human clinical studies. And then you have the phase 1, 2, and 3, with phase 1 and 2 mainly looking at the safety of the vaccine, and second at the immune responses, but not at really the efficacy against the pathogen. This is being done in a phase 3 clinical trial, and with over 10,000 people most of the time, sometimes over 100,000 people. This phase 3 is where we look at the efficacy of the vaccine. It is why it costs so much money, because of the number of participants that need to be included in those studies. And this is a lot of clinical site, a lot of people involved, of data management, and so on and so forth. So usually this is the period also that takes the longest. This is where also we are cutting time in those phase 1, 2, 3. How do we do it faster without cutting any corners? Is that even when a phase 1 is starting, we are investing into getting the phase 2 and 3 ready, which is not normally done. Normally we have to wait the end of the phase 1 to see if it is worth putting all the resources into a phase 2 and a phase 3. So in this case, a lot of time is being saved by cutting the time between those phases, by being ready before even having the result. That being said, I do believe that all the safety issues are being looked at with the same intensity, and there is no corner being cut here, but more in terms of logistic is being accelerated. After the phase 3, the vaccine, if successful, can be licensed, which means that it can be made available to the public. And one extra layer of protection that people often, that we will probably hear more soon, I hope, is the phase 4, which is actually monitoring of post-licensure availability of vaccine. So once the vaccine is being available to the public, or to at least subgroups of the public, then there is still monitoring. And we have seen vaccine in the past being removed from the availability to the public in phase 4, including more recently the dengue vaccine. So I wanted to highlight, using the Ebola vaccine, the importance of investing in vaccine. The Ebola vaccine that was initially developed in Winnipeg was an original idea of Heinz Feldman at the time that did the early work. And then there was a big team that joined ranks. This vaccine is basically based on an animal virus that does not cause disease in human, but that does replicate at low level and can kind of provide a photocopy of the Ebola virus to our immune system and produce in that sense protection. And so what's being done here is the outside shell of this VSV virus, which is vesicular stomatitis virus, animal virus, is being basically changed for the Ebola glycoprotein, just one protein of the Ebola virus, and the immune system sees this as an Ebola virus, but it's not. So this vaccine is now was licensed in 2019, late and early 2020 by Merck. And I think is a great innovation and proof of innovation and success in vaccination worldwide, with an initial funding from the Department of National Defense in Canada that believe in that vaccine, all the way to IDRC and others that have contributed to clinical trials in Europe and West Africa. These vaccines were generated with in mind the ability, what was needed basically. What did we need as a vaccine? We knew from going to outbreak and being in that environment that we would benefit greatly from a one-dose vaccine. So a vaccine that can afford protection after only one dose. You can see Heinz here on the left panel. These were very difficult environment, and they were very important to learn about what can be needed for a vaccine. By the way, on this slide, that is the lab in 2007 in Moika in the Democratic Republic of the Congo. You see two cooler-like boxes on the floor, top left. One of them had actually already the vaccine in it, and this vaccine was actively being developed and researched to make sure that not only protection was going to be afforded, that we could bring it to the field and it will be still capable of triggering an immune response. I'm just showing you a picture now to show you that these were not easy environment. Now with COVID, thankfully the environment is easier in many respects, but things can be done. And the support that was received by Frank Plummer and the Public Health Agency of Canada was extremely important to achieve this success. This slide is not for you to read through. It's just to tell you that it took time to develop that vaccine. But another vaccine also was developed at the same time from Winnipeg, together with the University of Pennsylvania and Philadelphia, is the chimpanzee adenovirus vaccine, which also reached phase 3 clinical trials and now is being used for COVID by many companies. Actually, when you talk about AstraZeneca as an adenovirus and many others, Johnson & Johnson is also another type of adenovirus, but this work also was influential for the development of new vaccines. So this is a list of all the clinical trials that were performed, mainly during the crisis of Ebola between 2014 and 16. When I will remind you, this was a crisis, but not to the extent of where we are now. This is a lot of clinical trials. As I told my student, if I would have proposed this in mice, probably they would have told me, no, you need to cut down, and that's too many lines and groups. But this was done, and it's to tell you that incredible thing can be done. This required an amazing amount of coordination. Another thing that came out of Winnipeg that I think is changing the way we can manage infectious diseases is the monoclonal antibody treatment that was called ZMapp at the time. This treatment that could treat infected macaques and later was administered to many humans, over 100, and shown to be, in macaques, could be saving the animals, is now being adapted to COVID, same approach, and the Regeneron treatment that we have heard more recently with Donald Trump being receiving that treatment is exactly the same approach. So monoclonal antibody attacking the COVID or SARS-CoV-2 virus as a treatment. And I think it's going to be a very interesting treatment to follow. And I would say that I'm quite optimistic that this treatment will work better even than the one currently being used with the steroid for ICU people. So this slide is, so what can we do in science? Well, we can develop tools to do safer, better, faster, and easier technology. Regulatory agencies apply laws, they cannot change them. And if we make their life easier, it means we can go faster to clinical application. And so how do we do that? We can have new platform. There's a lot of new vaccine approaches now with the adenovirus, the RNA, DNA. These are all new approaches to vaccination. A new administration device, a new innovative approach. I don't want to go through all this with the gene guns and tattooing and all this. I'm going to touch to the tattooing, but I wanted to show you and disclose for the first time this panel on the right. It's an HIV vaccine being developed in Canada, funded by STAG-IH. We have just, the first time I'm showing the data, that we have one of the vaccine, group 3, the green line, that is extremely promising. We have 60% protection in this group. And this is very important, by the way. It's one of the top 2 HIV vaccine in the world that we developed with $4 million and 2.5 years. The best other vaccine took over 10 years and 120 million US in the US by our colleagues. So we're very excited about this. We think that another 2 years on this and a bit more funding, we will basically be able to bring this up to 80% or more. This is a milestone, just to remind you, that 35 years of vaccination against HIV, and the progress have been very slow. So why is it important to innovate. It's also important because we need to develop all the tools we can to have a better healthcare for less. Basically, where every dollar counts. It's already the case. On the left side of this graph is a projection that was going all the way to 2014, and I'm showing you this. I thought it was quite interesting because the projection was actually quite on the dot. And on the right side is the cost per person that is current. It's actually a bit over 7,000 for 2019, $7,000 per person. The 2018, 2019, I've seen a growth in healthcare cost in Canada of 4.2, so above 4%. And so these innovation are also designed, and the goal is to bring that cost down or to get more for the same amount of money if you want. And this is very important. And how do we do this? This is a timeline of the Ebola vaccine. Sorry, it took basically 20 years. Because in fact, it started in 2001 with Heinz and was licensed by the FDA in 2020, the EMA in Europe in late 2019. So really 20 years. When we looked at new technology, here I'm just showing as an example, DNA vaccine, 3 years for an Ebola vaccine, 13 months for clinical approval. And then you see MERS, Zika, where we beat all the record with less than 7 months. From a vaccine on the screen to phase 1 clinical trials that did occur in Quebec City. But COVID now is incredible. 2 to 3 months, the RNA 2 months, DNA 3 months before entering. It's only phase 1, but it's already, you can see the progress that were made in the past 5 years alone. And what we need really is a vaccine that can make a difference in 3 months. A vaccine that can be released in 3 months, at least in subpopulation like healthcare workers. This is getting back to where we stand now. The lower left graph is on COVID-19. These are numbers from WHO. They are from yesterday. And you see a constant rise since the beginning of this epidemic. You see by region here, especially look at Europe. If you remember the first wave when it was quite difficult in Italy and Spain and other places, look at the second wave they are experiencing now. It is quite superior to the first wave. Unfortunately, we can kind of see the beginning of this happening in North America as well, in other regions of the world. There is rise, but not everywhere. Southeast Asia seems to be declining, which means also that a lot of countries are showing that you can control this epidemic, going back to basic of contact tracing, diagnostic, and so on and so forth. But one great news about this is look at the black line is the survival rate or mortality rate, if you want. And the black line substantially went down if you do the ratio per number of cases. And this is really important. So much, the number of people dying of the infection are lower, which means we are better at protecting the most vulnerable. We're better at protecting, everybody's better at protecting themselves. This is in Canada. The top graph is the number of cases. And the bottom graph is the number of deaths. And you can see again that despite the fact the number of cases is climbing, and the goal is to bring this back down, and a lot of provinces in Canada right now have added pressure on the virus with a lot of measure in place. And you see that the case fatality rate, so the mortality rate is lower. And this is really a good sign. It means that a lot of measures are working. It's not perfect. It's not 100%, but it's already a big difference. This slide is showing you in age strata. So who is more likely to die of COVID? This is nothing new. We are aware that the elderly are more susceptible to severe disease and death. But I'm showing this because I think it's very important. First, it tells you which population could be prioritized for vaccination. Second, if you look at the ICU, so intensive care unit, you see a disproportion, much less 80% and older compared to the deceased and compared to the hospitalized. And somehow it tells you also that a lot of the older people are dying actually in long-term care facilities or before reaching the ICU. And this we knew from we learned the hard way in the first wave, and now I think a lot of progresses were made, and that population is much better protected. So back on vaccine, the slide shows you the number of vaccine, very, very high. We are past 200 different vaccine regimen being tested and developed against COVID. let's focus on the phase 3. 9 vaccines made it to phase 3, which is incredible in less than a year. And some of them actually are due to report their data before the end of December. So it could be still possible that some emergency use authorization in some location could be happening to give access to the vaccine to some subpopulation. I think this is going to be quite interesting and important to follow. This is a list of the vaccine that have been secured by Canada for with contracts. So 2 RNA, Pfizer, Moderna, Johnson & Johnson with an adenoviral vectors, Novavax, Sanofi, protein, AstraZeneca, another adeno, and very recently last week, Medicago, the planned VLP. So, and to highlight is most of those vaccines actually are in the experimental, so to speak. And what I mean by that is there is no commercial product existing right now on the market. And this is important also because it tells you that we are redefining the tools that we have for vaccination currently. And this will impact the next dozens of years, if not hundreds of years. And this is, so we are right now really in an important time for vaccination. So what are the strand challenges and opportunities? The strand is that there is a lot of resources being put to accelerate timelines. There is a lot of novel and flexible technology development with increased coordination. And you've seen that, especially with the RNA, DNA, they have been moving extremely fast, faster than we have ever seen in vaccine. The challenges, as everybody knows, is time. Everything takes time. And I think one of the quote that I've given recently is that believing in rapid vaccine development is also believing and accept that it will take time. Other challenges that will be coming for sure are already there otherwise is manufacturing. The logistic, a lot of those vaccines must be kept at in very cold temperature. Vaccine uptake hesitancy, hopefully will not be too much of an issue. And of course the unknowns that we don't know yet. So let's see what may come up. For Ebola, it was the qualification of the clinical production plant. So the manufacturing building, it was a new vaccine, and that took 2 years alone. So these are sometimes things that we cannot see coming. The opportunities are, again, I think very, very important that we are reshaping the field of vaccine and future ability to quickly respond for generations to come. I'm going to show you very quickly some technology that we've been advancing. So this is a tattooing machine, off-the-shelf human tattooing machine that was modified by the engineering department at the Université Laval. And it's now actually a vaccine injection device for any vaccine that is in liquid form. It is with an array that you see of 11 needles, which also exists for human tattooing. And it injects vaccine into the intradermal space, so in the skin, if you want. This is the best place to give a vaccine, but it's very, very challenging to do this with a one needle, and you're very much restricted to a very small volume. Instead here, a very low-tech technology, so to speak, that can administer large doses of vaccine. And what we see is that with 1 or 2 doses of vaccine, we can have the same protection with commercial vaccine that otherwise will need 4 or 5 doses. So 1 to 2 doses to do the work that 4 or 5 doses otherwise must be given in the muscle. Another, I think, and here this is a little bit my baby, so you'll allow me to present that. This is a not-for-profit. It's called GUARD, a Canadian not-for-profit that goal is to bring prophylaxis and therapeutics product that do not have a traditional commercialization pathway. Basically, what it means is that a lot of vaccine and therapeutics are not a good product for companies because they are not money maker. We are advancing those products. We are doing it in a context where profit is not allowed. Basically, it's not within a for-profit environment while creating good job. So it's good still for everybody that works for GUARD. We're paying very good salaries, but no big millions to anybody and no big profit. The profit always reinvested. We have several programs including very proudly a surveillance program in Africa that is also building capacity. This is funding with IDRC and GAC. Also DARPA. DARPA is the US government Department of Defense that is funding a phase 1 clinical trials through GUARD of a latest generation Ebola vaccine and other projects that you can see here. And I will add this, Guardrx is operating 7 labs right now in Northwest Territory, Nunavut, and North Quebec. We have performed over 54,000 tests at no cost to the government, and we have detected over many cases, but none of those sites have seen outbreaks of COVID. We have kept every single site secure and free of COVID. And even if there will be one tomorrow, I can tell you this over 5 months to us is a great sign of success to bring those workers back. These are, a lot of those are mine site, actually, very crowded community in small spaces where eclosion would be explosive. And we have kept all these sites secured, no COVID. And we think it's a very important aspect of getting back to work in a safe environment. And, you know, the not-for-profit is not against for-profit. Actually, this company was born from GUARD. It is a for-profit company that sells mobile labs that you see the design here. There's many across Canada and many were exported outside of Canada. They are COVID now, sold as COVID lab. They are pre-equipped with COVID testing. And it is 20 years of experience in lab, mobile lab capacity that were put into the design of this. And I have nothing to do with it now, but I'm very, very happy to see it thrive, basically. And there's one actually at the airport in Val d'Or that has of now done an extremely good job at capturing people that are traveling. This is a very busy slide. I'm not going to go through it, but I just wanted to tell you, and if you have time, take a screenshot. This was developed by the students at McGill University and University of Montreal in immunology and microbiology. This is their view of the strengths, challenges, and opportunities against COVID-19. And you see, they see a lot of opportunities, just the sheer numbers of words there. And this was very refreshing to develop that slides together with them last week. And this baby on the right is, by the way, not what we hope to achieve with the tattooing machine and vaccination, and we're not targeting right now pediatric vaccination. Of course, the tattooing machine can inject a vaccine colorless, but doing this for animal health, and vets are pretty excited about this, that they can also mark them as they do anyway. So wanted to thank, I'm not gonna go through all the names, but these are terrific young scientists in my team, and all the collaborators on the right. There's a lot more. I just selected the main one. A huge thank you. Without funding, this will not be possible. And huge thank you to IDRC. I think one of the most amazing funding department, federal level Canadian. And I'm not saying this because I know Jean. It is what I think. And also CIHR, quite impressive, amazing actually structure in Canada for scientific research. Thank you so much, everybody, for your attention.
Bonjour à tous et à toutes. Premièrement, au nom de l'honorable George Fury et le président du Sénat et l'honorable Anthony Rota, le président de la Chambre des communes, je voudrais remercier tous les honorables députés, l'honorable sénateur, les intervenants et tous les invités distingués pour votre participation du premier événement virtuel des têtes à Papineau. In this 25th anniversary of PAGC, we public office holders, along with scientists, academia, and advisors to public policy, we find ourselves confronting a health crisis on a scale that most of us have never encountered. And while certainly COVID-19 is taking our focus these days, we're also mindful of the fact that the role of empirical and dispassionate scientists is as important as ever on areas like food security, on humanities and security in general, and the environment, to name a few. And it's why we owe so much to those 21 national research organizations that really comprise PAGC. And it's with this in mind that I know you'll join me in noting the sponsors of today's event, and for all the rest for their ongoing efforts in providing scientific insights for our work in the public forum. Today, Dr. Kobinger gave us some incredible insights into what is surely the most anticipated scientific discovery of our time, a vaccine for this pandemic. Understanding the path that a vaccine development takes historically, and the journey that the vaccine for COVID-19 will invariably take is invaluable. C'est pourquoi je vous rejoins d'exprimer nos remerciements de sa présentation aujourd'hui. Merci, Dr. Kobinger, pour nous informer concernant ce sujet très pertinent et veuillez accepter notre gratitude. Merci beaucoup.